We're delighted to welcome onto CVCT Cardio Brief blog, Professor Paul Armstrong from the University of Alberta, who's had a lifetime of doing clinical research in a variety of aspects of cardiology to try and make lives of patients with heart disease better. And today he's going to talk to us about what, to me, is a completely new class of drugs in chronic heart failure. So Paul, can you tell us something about this? Can you start a little bit with the mechanism of how the drug works? Because it's completely new to me.
So thanks, Darrell. The drug in question is called Vericiguat. It's difficult to spell and to pronounce, so you need some practice. But it is a cousin of a drug called Vericiguat that was fabricated in the laboratories of Bayer in Germany for the treatment of pulmonary hypertension. And Vericiguat is a once-a-day drug, and we recognize by way of backdrop that heart failure is a pro-inflammatory state and one where nitric oxide is deficient. And as a result, there's endothelial dysfunction and failure of the intracellular energy mechanics to generate cyclic GMP, which in turn drives energetics, affects smooth muscle and cardiac function. And so what Vericiguat does, because it's a soluble guanylate cyclase stimulator, is it actually penetrates the cell and directly stimulates the GMP and also restores sensitivity of nitric oxide, such as is residual exists. So it essentially has a bimodal action, one directly stimulating and two enhancing NO to drive the cellular machinery in a more energetic and productive way. So that's the background.
So although it may sound a little bit like the cyclic AMP agents in the past that have been positive inotropes, actually it is different. It just has a few words in common in the mechanism.
Correct.
It goes into the cell and works on the soluble molecules rather than a membrane receptor.
And we've had two indirect common drugs that you would recognize that have worked on this pathway but not been successful. One are the nitrates, which of course simulate NO, and the other are the phosphodiesterase inhibitors, which prevent degradation of cyclic GMP. And so this guy then gets in between and is a direct SGC stimulator.
Fantastic. So what was very interesting about this study, to me, as you know, we're running a bit of a course on how we design clinical trials, is that you didn't design it like most trials, whereby the sample size calculation was based entirely on the primary endpoint and aiming to get a good but not astronomical power on the primary endpoint. Instead, you set it for a very high power on the primary endpoint. And as I understand it, so that there was also a reasonably good power on the secondary endpoint. Is that right? Can you take us through your reasoning in that?
Yeah. Well, it was obviously, this was a partnership between our Vigor Center in Alberta, the Duke Clinical Research Institute, and the two corporate sponsors, Bayer, which discovered the drug, and Merck, which partnered with Bayer to develop it. And so because they were going obviously for a labeled indication, the trial was deliberately overpowered on that primary endpoint. We knew that we had to have 787 deaths, and we reckoned we would have somewhere in the neighborhood of 1,480 or 1,500 overall events. What surprised us was that, in fact, we had much more heart failure hospitalization than we expected. And so, and we targeted a population knowing that they had to be on good baseline therapy. We wanted a number of events, and we assuredly got them with a very sick.
Am I right in saying you had a higher ratio of hospitalizations to mortality than you thought you would?
Yes. Yes.
And your assessment was based on experience of other patients in that category
correct.
of illness?
Correct. And phase 2 data, the phase 2 data that used the surrogate endpoint, we knew that natriuretic peptide was important as a criteria in terms of sick patients who had worsening heart failure. We wanted to be sure we had the right population and enough events to address the hypothesis.
Wonderful. So let's now turn to look at the primary endpoint results of this trial. Can you take us through the highlights of this?
Right. So the first thing to say is that the placebo event rate was extraordinarily high, of the order of 38 or 39% at 12 months. And Vericiguat reduced that hazard ratio 0.90, confidence limits 0.82 to 0.98 with a p-value of 0.019. So the relative reduction was less than we had anticipated, still significant, but the take-home message was that the absolute reduction was actually extraordinary. It was 4.2 absolute event reduction per 100 patient years, which is compares very favorably to other conventional trials. So here we see this important relationship between relative and absolute risk reduction, which ends up with a number needed to treat of about 24, which is quite realistic.
Remarkable. Your original sample size calculation was based on a much lower total number of events, and you got many more events than that. So let's look at the components that made up the graph that we were just looking at.
So in the left panel of this slide, we can see that cardiovascular death did not reach statistical significance. The directions were in the right direction, as it were, but heart failure hospitalization, we did achieve a significant treatment effect concordant with the primary composite event. The other thing to say about this trial is that we pre-specified, after discussion with the FDA and based on the background phase 2 data, that we would look at the quartiles of natriuretic peptide. The natriuretic peptide criteria for getting into this trial were very high. And as you can see, the first three quartiles, first of all, perhaps a perspective here, the median natriuretic peptide here was about 40% higher than any trial in play. And you can see that there's a striking interaction here, which actually, although we were not allowed to report it in the New England Journal of Medicine, the interaction p-value is p less than 0.001 on this interaction.
Wow.
And so this obviously has led to a great deal of discussion. And indeed, we have gone on to explore that as a continuous variable. But we see actually a reduction in both cardiovascular death and heart failure hospitalization in these lower three quartiles. And as we look at the baseline characteristics of this upper quartile, as you would expect, they're older, with worse renal function, more comorbidities, and just tougher nuts to crack. So on the one hand, this is very interesting. On the other hand, of course, we have to live with our primary endpoint, which was pre-specified. But the fact of the matter is that this, I think, leads us to an exploratory pathway with our biomarkers and other elements vis-a-vis natriuretic peptide, which, as you know, is considered to be a pretty valuable prognostic marker, a pretty effective surrogate marker. And indeed, as you look at sequential data, which we haven't yet reported, it's clear that the lower you go, the better you are. And that's true even in the placebo group. It just happens that there are a lot more patients with Vericiguat that get lower BNP than placebo. So that's kind of where we are.
So these are pre-randomization values, are they?
Yeah, yes, baseline.
Okay, really interesting. Can you tell me what you think the timeline might be for us to be looking to apply this drug more generally? Are you planning other trials? or do you think this trial on its own should get it into general use?
I think this trial it's under review by both FDA and European Medicines Group, and we anticipate that we'll get some news on that in the first quarter of 2021. So I'm pretty confident, based on the initial reviews, that this will indeed lead to approval. But of course, as you correctly anticipate, it's not the end of the story. And the question will be, if one used this agent in patients who were less sick, for example, patients such as were studied in DAPA or Paradigm, and one were able to reproduce the natriuretic peptide data I've just shown you, then indeed it would be, I think, look at very favorably. But bottom line is, it's a once-a-day drug, very safe, good adherence, no need for laboratory monitoring, potassium or renal function, and I think as such.
So that's a very important point, because we often come up against it with renal function. In fact, on your nice diagram, are you able to indicate whether you think it works primarily through the heart or primarily through the peripheral vasculature in this?
So we have sub-studies in the cooker, and the next time we talk, I hope to be able to answer that question, but I'm afraid, although I don't like coming down with ambiguous answers, I will say we're working hard on answering that question. And
I think it's absolutely fine to be ambiguous on it, because you know, part of science is knowing what you do know and knowing what you don't know, and that's what we do for a living, is try and separate them. Thank you very much, Paul. I know you've got to rush to another call now.
Pleasure. Thank you. Bye-bye.
Bye.